Yes, so hello everybody, my name is Nele Villabruna and I did my PhD at the Erasmus Medical Center in Rotterdam, the Netherlands, and I will present my work on comparisons within the ORKG grant 2022. So first, some background on the topic. So as has already been mentioned, my grant was awarded by the ZB-Med Information Center for Life Sciences, and this was specifically meant to target the topic of microbiology. And since I did my PhD on viruses, I focused on viruses rather than other microbes and bacteria. And I did most comparisons on SARS coronavirus 2, because of course this is of interest at the moment for a lot of people, and also on noroviruses, because this is the virus I focused on during my PhD. So my comparisons were broadly grouped into three bigger questions. So the first one was where viruses are coming from. And here I specifically looked at whether viruses can jump from animals to humans or vice versa. In the second group of comparisons, I then focused on viral and host factors that impact the disease severity. So, for example, if the amount of virus particles in infected people impacts how severely ill these patients become. Or another example is how the different arms of the immune system protect you from severe disease from different viruses. And then the third group was where specifically in the human body these viruses infect. And this is also the comparison that I will focus on now. So this is what is the tropism of SARS coronavirus 2 comparison. So here I looked at papers that focus on which organs in humans are infected by this virus, and more specifically which cell type. And this is important information for several reasons. One is to better understand the symptoms. So if you think of SARS-CoV-2, we think of it mostly as infecting the lungs. And that means you also expect symptoms that are associated with the lungs, like coughing or pneumonia. But then if you see symptoms that are outside of the lung, for example, the brain, then you would need to know if this is because the brain is actually infected or if these symptoms are not correlated at all with the infection with this specific virus. Then it's important for drug development, of course, because you need to know if you develop a new drug, which organs need to be targeted. And finally, it's important to understand and maybe to prevent transmission, because if you think that the virus is only transmitted through the airways, then you would try different methods to inhibit this transmission than if you know that, for example, it's also transmitted by stool of infected individuals. So for this comparison, I did 69 contributions from 33 papers. And as I already said, for SARS-CoV-2, of course, we think of the lungs as being the most common target. But because now we have like 3 years of studying this virus, and we have a lot of infected patients, and we have also seen that a lot of other organs are impacted and damaged. So the best known is the gastrointestinal tract. So many people also have an infection in their intestine, but there are also patients who have neurological symptoms, who have infections of the heart, the eye, or several other organs. On the right side, it's the same information, just a different way of showing it. So also here you see that the lung is still the most important organ that is targeted, but a variety of other organs and body parts can be infected by this virus. In addition to the organ, I also extracted information on the cell type, because of course most organs will be comprised of different types of cells, but often viruses do only target a specific cell types rather than just all cells in general of an organ. So the information I extracted first on the host was, of course, which species. So I focused mostly on human studies and not so much on animal studies. Then I extracted the age, which symptoms these people had, which sample was taken. So this was mostly biopsies, and then also how many participants were included in the study. I also looked at the approaches and methods. So the first two, the immunofluorescence and in situ hybridization, are basically just staining methods for certain viral proteins that can be detected in these biopsies. The RT-PCR is just to look for genetic material of the virus, and electron microscopy is really visualizing the virus particles themselves. And these were the most common organs that were mentioned in these papers. So of course, the lungs, the intestine, kidneys, and several others. And the cell types I put here in two categories because there are some cell types like the endothelial and the epithelial cells, and these are not organ specific, but are just found in several organs, while others, such as the optic nerve, is very specific. So this one is only found in the eyes, for example. So I will switch to the comparison itself. So this is how it looked like then. So I had the research problem, of course. So what is the tropism? Then the approach, and this was, of course, depending on the method that was applied. So whether they looked for replicating virus or just some viral components. Then the date, the species, the sample size, age, the symptoms. So these samples were often taken from diseased patients. I have here the methods where I also added the description of the methods and, if possible, the link to the Wikidata page. And then in the end, the results. So here the organs where I also tried to add a description normally and a link, as well as the different cell types. Yeah. And in the bottom, it was just whether these organs were infected or not. Yeah. So with this, I end. And of course, I would like to thank you for your attention, and the ORKG and the ZBMed for allowing me to add these contributions.